Okay, and then without further ado, I'm going to turn it over to John Weil, research scientist with NJDEP's Bureau of Freshwater and Biological Monitoring.
Thank you, Beth, and thank you for giving me the opportunity to present some of the results from our fish IBI and environmental DNA monitoring in the Paulins Kill watershed from 2022. Before I get into any of those results, I just want to start off by giving a little bit of a background on our New Jersey Fish Index of Biotic Integrity and our environmental DNA monitoring, in case folks were not familiar with that. A fish IBI is used to compare a fish assemblage of a particular stream to that of a least impaired stream from the same size, from the same ecoregion. So fish are relatively long-lived, and therefore they're good indicators of stream health and long-term trends. Fish reside in the stream year-round, so therefore they're exposed to any water quality or habitat stressors that may exist. We've been conducting IBI monitoring in the Paulins Kill watershed for over 20 years, so we have a lot of historical data in the watershed, but 2022 was the first time we were in there sampling since the Columbia Dam was removed. So again, our fish IBI was established in 2000, and it uses multiple attributes that we call metrics of a resident fish, amphibian, crayfish assemblage to measure the health of streams. Fish IBIs are an effective tool for monitoring long-term health of a stream. In New Jersey, we currently have 3 IBI programs. In the southern part of the stream, we have a Southern Fish IBI that only right now monitors those streams in the intercoastal plain outside of the Pinelands, anything draining into the Delaware River. And we're currently embarking on a process to revise those metrics to also include Atlantic drainage streams. My coworker, Brian Henning, is also working on a project to evaluate whether we can come up with a bioassessment for the Pinelands. In the northern part of the state.
I'm on the
I think somebody may not be muted. If you can mute yourself. As I was saying, in the northern part of the state, we pretty much cover all streams that are non-tidal and wadable. So in those very small streams, we have a headwaters IBI that utilizes fish and crayfish and amphibians as bioindicators. And then the counterpart to that is our northern fish IBI, which utilizes just fish in wadable streams that are over 4 square miles in drainage. As you can see from this box plot over here of our headwaters IBI and our fish IBI, comparing overall scores between least impaired, intermediate, and most impaired sites, we have very good discrimination amongst stressors for both indicators. So several years ago, Brian Henning and I took on a multi-year process of developing new metrics for our northern IBI and headwaters IBI using a structured approach that was developed by Bob Hughes and Tom Winier, 2 well-known established fish experts from Oregon. And what this process does is that you select metrics from ecological categories of a fish assemblage, put them through a series of rigorous tests, and any metric which fails any aspect of the test is removed. So you end up with a series of metrics that are very sensitive to the type of stressors that you're seeing in that ecoregion or state. All of our methods and analysis and results were all reviewed by an expert panel made up of state, federal, and academic biologists. Brian and I also published this work in a peer-reviewed journal in Ecological Indicators. If anyone wants more information, I can certainly get them a copy, or you can look up Ecological Indicators to try to get a copy. These are the 8 metrics that were selected for our Northern Fish IBI and the ecological categories that they represent. So in our stream flow category, we have the percent rheophilic species. These are fish which prefer flowing water. And as you channelize a stream, as banks are eroded and stream flattens out, and you lose some of that flow regime, it turns into a slower, sluggish stream. These fish are impacted and will tend to decline. Our thermal category is represented by our percent cold water species and non-tolerant cool water species. So as riparian buffers are removed, as overhead covers are removed, the streams flatten out, you no longer have those riffles and those cascades that deflect sunlight. Streams increase in temperature and these fish are affected and tend to decline. Our trophic category is our percent generalist feeders. So any healthy fish assemblage should be comprised of specialized feeders, those which feed on insects, those which feed on other fish. And when you tend to get into generalist feeders, which will feed on anything, that's typically an indication of stress. So these generalist feeders will increase with the stress in the aquatic system. Our tolerance index is a measure of sensitive, intermediate, and tolerant fish. As you increase any kind of stress in the system, you lose those tolerant, intermediate fish, and you tend to get more tolerant fish that remain. And so this tolerant index will increase with any stress. Our reproduction category is our percent lithophilic species. So these are fish species which require clean natural substrate in order to spawn. As you increase sediment deposition, fine sediment fills in a lot of these interstitial spaces. These fish can't reproduce and will tend to decrease. We have 2 composition metrics. The percent cyprinid species metric is the number of minnows. So in any healthy northern site, the bulk of the assemblage should be comprised of minnows. They're one of the most abundant fish that we have, fish taxa groups, and any kind of impairments, you tend to see these minnow species decline. And then the percent dominant 3 taxa, when you go to a healthy fish assemblage, you should have very good diversity. You should have even numbers of multiple taxa. You should not have an assemblage which is dominated by just a few taxa. This is an indication of an impairment. And then our last metric that we utilize is our percent benthic insectivore species. These are fish which live on the bottom, feed on insects. Again, any kind of siltation in the stream is going to impact these fish' ability to survive and then also feed. This shows our Northern IBI ratings ranging from excellent to very poor and the categories and scoring for each one. Of note is our poor and very poor categories. So any site which scores a poor or very poor is considered impaired for aquatic life use. And it is listed in our integrated report as an impaired site. This is an example of our data reports that we post on our website. So this page, along with a second page, which provides the species list of all the fish, amphibians, crayfish that we collect at a site, is posted on our website. It provides the site location, the date it was sampled, the overall score and IBI rating, the rapid bioassessment habitat score and rating, our field chemistry measurements, our nutrient measurements, and then each one of these individual metric scores, all these are averaged together to get the overall IBI score. And then we'll typically write a summary on that site, just describing what we're seeing at the site, if it's anything in the assemblage or the overall score or habitat or water chemistry has changed since the last time we monitored that location. So now that we've talked about what metrics we use, what an IBI is, the way we go about collecting our actual fish is through electrofishing. So for our northern IBI sites, we use a combination of either multiple backpack, electrofishing backpacks, or we use a tote barge. What this does is puts a mild electric current into the water, stuns the fish. We're able to net them, place them in buckets, and then live wells allow them to recover. And then we'll come back and we'll ID all the fish. We'll check them for any kind of external deformities. We'll measure game species, and then we'll release them back into the stream. So this video shows us shocking a white sucker. You can see it was sort of attracted towards the probe by a process known as galvanotaxis, in which the muscles contract and force it to actually swim towards the electricity. And then we're able to net that fish and allow it to recover. So in 2022, we had a number of monitoring goals. Again, the first time we're returning to the Paulins Kill watershed to do any sort of IBI monitoring. So we wanted to document if there are any changes in the overall fish assemblage at fixed locations. We wanted to also see if there were any changes in our IBI scores at these locations, document the presence of migratory species. And we also were looking from an eDNA standpoint, document the presence of American shad throughout the spring and summer, see if we could document American shad spawning, and then also hope to find young of the year American shad in the watershed. So this map shows the location of our monitoring for our IBI sites in 2022. Most of these sites are all northern IBI sites. We have this one headwater site up on a tributary to Blair Creek. The rest of those are northern IBI sites. The color indicates green is an excellent score, yellow is good, orange is fair, and red is poor. We have one site out here, Phoebe 128A, which scored poor. This is a site which is just downstream of the hyperhumus complex, which is likely causing the impairment due to the unique water chemistry and habitat. Of note, in our monitoring, we did have 6 sites that had significant increases in American eel collection. American eels are a catadromous species in which they live in fresh water and they return to the ocean to spawn. So since the Columbia Dam removal, we had some increases in American eel. In addition to that, for the first time ever, we detected American brook lamprey and sea lamprey. So American brook lamprey are a sensitive taxa that live their entire life in freshwater, whereas sea lamprey are an anadromous species which live as a larval stage, we call it amicetes. They live in the sediments for anywhere from 5 to 8 years. They emerge from the sediment, they out-migrate out into the ocean where they transform into a parasitic phase where they'll actually latch onto the fish and feed on fish. And then as they mature, they will return back to their natal streams to spawn. Sea lampreys have kind of gotten a bad rap from some of their introductions into the Great Lakes, Lake Ontario in particular, where they're a non-native, they're considered invasive. They don't have any access to the ocean. But here in New Jersey, the Delaware River, they are a native species. They're a good indicator of water quality and habitat and substrate, and obviously connectivity. As I said, we typically don't collect adults, either American brook lamprey or sea lamprey, as they've spawned and died and are no longer in the system by the time we're electrofishing, but we collect the larval stage, the amicetes. If we look at the map, our Phoebe 55 site, which is on the main stem Paulins Kill, we collected both American brook lamprey and sea lamprey. And then, very interesting enough, at our Phoebe 001 on the main stem Paulins Kill and Phoebe 81 on Pond Brook, we also collected sea lamprey, which is interesting because they are both sites that are upstream of Paulins Dam. What's interesting about sea lampreys is they do have some ability to get over some lower dams under certain conditions, certain flows. So as adults are migrating upriver, if the flow conditions are right and the dam is low enough, they will actually suction onto the side of the dam and they can actually make their way over some dams. So based on our 20 years of monitoring, it would appear that Columbia Dam was an obvious barrier to these migratory fish that they could not get over. But it appears as if under certain flow conditions, at least some adults are able to move upstream of Paulins Dam. If we look at overall migratory fish abundance at those sites that we've been monitoring for more than 20 years, the two species we're talking about here are sea lamprey and American eel. Over the prior 20 years, the average abundance of these migratory fish averaged around 5% of the overall catch at each site. Compared to this past year, we're up over 14% of the catch was comprised of migratory species. It would seem pretty obvious that this is a result of the removal of the Columbia Dam, and I would expect these results in migratory fish abundance to increase as the Paulins Dam is also removed. So like I said, we do not typically collect adult lamprey, but while we're out this spring collecting environmental DNA samples, we did come across several spawning adult sea lampreys on the main stem Paulins Kill. So this is right at Footbridge Park. We noted a number of spawning adult sea lampreys. You can see they're latched onto a rock and then they intertwine and go through the spawning process. Moving on to our environmental DNA sampling. So this was a project that we piloted back in 2021, and we were looking to expand on this in 2022. What this involves is either collecting a water sample, or in the case of what we were doing in 2022 is filtering a water sample. That DNA is then collected on a filter that we provide to a laboratory for analysis. We were working with Rutgers University to perform all the analysis and DNA extraction. Our field sampling methods involve using either one of these backpacks that we have. We also have a couple of stream side packs that we use to filter the water. Basically, we're moving in upstream, keeping the filter end out in front of us to make sure we're not contaminating the sample with anything coming off of our equipment. Inside this filter housing is the actual filter. And this is a design by Smith Root in which this is a self-desiccating filter, which is important. The key to not losing any DNA is making sure that filter is dry. Any moisture in there promotes microbial activity, which will actually break down any DNA that's on there. So once we collect a sample and it's dried, it goes into a foil envelope, then into a Ziploc bag. It's put on ice till we return to the lab where we can put it in the freezer. And then after we go through several weeks or months, we get those filters to Rutgers University for the analysis. From our standpoint as fish biologists, this was a new process to us. We do a lot of fish monitoring, and with that, fish DNA sticks to everything. So our waders, our boots, our equipment, our vehicles, our laboratory, like everything has fish DNA on it. So Rutgers was very helpful in giving us the protocols for decontamination, since DNA easily washes off with DI water. So we had to take a lot of precautions. Any equipment we could, we sprayed with 2% bleach, which destroys DNA, and then making sure we're washing everything down, our waders, boots, all of our equipment in DI water, and then changing nitrile gloves. We go through a lot of nitrile gloves every time we're out doing sampling. So once Rutgers receives these filters, they can extract and amplify the DNA off of the filters. So every fish species has a 200 to 250 base pair sequence of DNA that's unique to that species. So for us, Rutgers was performing two types of analysis. They're performing a qPCR, in which they're specifically looking for 183 base pair target sequence of DNA for American shad. And then also when we're out collecting our eDNA samples, they're also using metabarcoding. And what this is comparing the DNA that they extract off of the filter to a library of DNA sequences for fish in your areas. So qPCR is very good for detecting rare fish, fish in low numbers, and a targeted fish. Whereas metabarcoding is very good for detecting an overall fish assemblage, but not as good at detecting those fish which are in low numbers or rare. So our target for qPCR analysis was American shad, an anadromous species that lives in the ocean, returns to the natal streams to spawn. It is nicknamed the founding fish by John McPhee in his 2002 book. They're an historically important migratory fish species for Native Americans, early colonists. They're reported to have sustained Washington's army during the Revolutionary War. But the eastern U.S. runs have been in severe decline as a result of dams, water pollution, and overfishing. One of the issues with American shad is they're classified as a forage fish species. So they're not protected under the Magnuson-Stevens Fish Conservation and Management Act, like many game species such as striped bass. A number of states like the Chesapeake Bay and its tributaries have enacted a moratorium on inland recreational and commercial harvest. But unfortunately, this has not resulted in an increase in the population in many of these systems. It's thought that some ocean fishing, collecting shad as bycatch, is having a big impact on the population, in particular some mackerel fisheries that aren't heavily regulated, and even the bycatch is not well documented. Now we know shad have been caught by fishermen accidentally, have been observed in the Paulins Kill, but there's a lot we don't know. We don't know if they're actually spawning, how long they're staying in the Paulins Kill, if there's adequate rearing habitat for juvenile fish, what the predation rate is in the Paulins Kill. So there's a lot we don't know. Pennsylvania has done some extensive studies on American shad in the Susquehanna, the Schuylkill, the Lehigh rivers. And through some of their tagging studies, they note that typically Delaware River stock of American shad typically do not stray very much. So what that means is they'll tag larval fish when they release them into the water. They're imprinted with the chemistry from that system. And when they return in 4 to 7 years as adults, they're going to go back to that system. They don't accidentally go into the wrong tributary. So based on that information from Pennsylvania, if American shad have been blocked from migrating into the Paulins Kill since 1909, when the Columbia Dam was constructed, why would they all of a sudden just return to the Paulins Kill? Now, I do believe that they have returned and have been somewhat restored to the Paulins Kill. I do believe that they're spawning, but I think it's also important to have good data to back up these notions and to promote the positive outcome from the Columbia Dam removal, so that can be used as a success story for future dam removal projects. So for 2022, our DNA American shad sampling, we started weekly sampling on the Paulins Kill in mid-April. We had hoped to get out there a little bit earlier, but some high flows prevented us from getting out. We continued weekly sampling into the summer for juvenile shad. We also conducted some spot electrofishing and netting at targeted habitats in the spring and summer for larval and young of the year American shad. Overall, we collected 210 eDNA filters on the main stem Paulins Kill. All of our field and laboratory blanks were clean. So each day we go out to collect a sample. We collect a field blank at the start of the sampling event for the day and at the end to make sure we're not accidentally contaminating that filter. And Rutgers does the same thing in the laboratory when they go to analyze. They run a laboratory blank to make sure there's nothing in the lab that's contaminating that sample. Overall, 84% of the filters that we collected resulted in positive detections for American shad, which is pretty good. So there's a series of maps which provide some information on how we're detecting shad, the movement of shad. These are our monitoring locations. eDNA 701 is our downstream location below the former Columbia Dam, just upstream of the confluence with the Delaware River. 702 is off of Station Road. 703 is Sipley Road, also happens to be our IBI monitoring location, Phoebe 55. And then we have eDNA 704 at Footbridge Park, about a mile downstream of Lina Dam. So the way that the map works is I go through is each time we go out and collect DNA at a station, we collect 3 filters. So if we get a positive detection on one of the filters, a fish icon will show up at one of the stations. And if Rutgers, when they run the analysis, if they get a positive detection, then they run that sample in replicates, they run additional replicates. So for each hit, they'll run 3 replicates. So if one of the filters has a hit for 1 replicate, it will show up as one of these smaller fish. If 2 of the 3 replicates have a detection, be represented by a medium fish, and then a larger fish for a positive detection on all 3 replicates. Over in the right-hand corner is a graph which shows the mean DNA concentration per liter of water filtered for each of the stations. At the left-hand side is our 701 downstream location, and then upstream is 704. So this was our very first sampling event on April 14th. We already have shad detections in the lower sections of the river. So the lower 2 stations, we already are detecting shad on all 3 filters. 2 of the 3 replicates for a couple of the filters, 1 of the 3 replicates for the other. Just starting to detect shad up at our 703 monitoring station. But you can see the bulk of the DNA is still at the downstream locations. As we move into April 25th, now we're detecting quite a bit more DNA at our downstream locations. 701, we have all 3 filters and all 3 replicates. We're detecting shad DNA. Just starting to detect shad DNA at our upstream location at 704 at Footbridge Park. The bulk of the DNA is still at the downstream location. As we move into May 2nd, we're starting to detect DNA throughout the river now, quite a bit more at our upstream locations, all 3 filters, 2 or 3 filters at our most upstream location at 704. And then on May 9th, this was kind of interesting sampling event. We had a thunderstorm event the night before. The river was rather turbid, and as we moved upstream, the turbidity actually got worse. So by the time we got up to 704, we weren't able to filter very much water. The filters clogged pretty quickly with sediment that was in the water column, and we just don't think we got a very good sampling event. By the following week, May 12th, we're kind of back to where we were before, where we're detecting quite a bit of shad DNA, even at our upstream location here. And as we finally moved into May 23rd, now we have shad DNA detections on all filters, all replicates throughout the river. And this was kind of an interesting day because this was the most DNA we had collected of the entire summer. And it was also the first time in which we've sampled in which the river temperatures were over 65 degrees. So optimal spawning for American shad is 65 degrees. So based on the results of our DNA sampling, what we're seeing in the river, we think that this is probably right around the time that shad are actually spawning. So on the same day, we noted a number of schools of American shad. This is just downstream of Paulins Dam. This is most likely a school of American shad that are either spawning or just post-spawn. And as we get into the end of May and early June, we just have similar detections where we're picking up quite a bit of DNA throughout all stations. Again, into June 6th now, still quite a bit of DNA. June 13th. And as we get into the middle, end of June and into July, we begin getting these sort of peaks at different stations. And some of this, I believe, is juvenile fish that are now up in the water column. We're detecting their DNA a lot easier. And we're also seeing movement of young of the year juvenile fish out of the system. So we're starting to see these peaks now and some slight decline at our upper site, which seems to be an outmigration of these young of the year schools. So on July 7th, we have this big peak at station 702. And now this is the first time we start seeing quite a bit of a drop off at our upstream location. So now we're still detecting shad DNA on all three filters, but now we're down to 2 filters only have 2 or 3 replicates, and 1 filter has 1 or 3 replicates and quite a bit less DNA. And I believe this peak is again a school of probably juvenile fish that are moving out of the system. As we get to July 11th, now we have this big peak at 702 or 703, my apologies, at this station, but now we're no longer detecting any shad DNA at our upstream location. So this sure seems to be like those fish that were upstream are now moving down. We're picking them up here at 703, and also we have this other peak all the way down at the last monitoring location. So we get to July 18th. Now we're still not detecting any shad DNA at our upstream location, only collecting DNA from 1 filter, 1 to 3 replicates at our 703 location. We have this spike down at 701. As we finally get to our very last sampling date on July 26th, we're no longer detecting any American shad DNA at our upstream sites, and we're starting to see some declines in our downstream, where we're only getting 2 or 3 replicates on a couple of filters. And this graph shows the overall across the entire time period, the average DNA concentration as the green line. Average DNA per liter filtered. This blue line indicates the mean water temperature. Here's our 65-degree optimal spawning water temperature. And we have this peak, the largest amount of DNA we collected on that May 23rd date, which I'm pretty sure is an indication of spawning, where you are not only picking up DNA from adults, but you're picking it up from eggs and milk that's in the water. In addition, we have these 3 spikes. This first spike here is likely indicating the first big pulse of American shad in the spring, moving into the Paulins Kill. And then we have this one later on in the summer in July, which is likely indicating schools of juvenile fish that are now in the water column and are now out migrating. So in 2023, we want to continue with some of this work, hoping to capture the beginning of the run and the end of the run a little better. So we initiated weekly sampling on April 4th. So we started 2 weeks earlier. So hopefully we can capture when shad are just moving into the Paulins Kill. We plan to continue weekly sampling into August to try to capture when these juvenile fish are have completely left the system. This year we initiated larval drift net sampling. So on May 23rd, we performed our first drift net sampling event. Basically, we anchor a drift net in the evening at our 3 upstream locations, 702, 703, and 704. We come back the next morning and collect the net, collect the sample, preserve it, and hopefully through this we can detect some larval fish and American shad in the sample. We plan to conduct some spot electrofishing and seining at targeted habitats this summer for young of the year. And just from a DNA standpoint, we plan to continue collecting eDNA at our IBI locations in conjunction with electrofishing. With that, I just want to thank the entire fish crew for their flexibility in assisting with this project. When we take on a new project like this, we still have all of our routine work to complete. So sometimes it gets a little bit hectic. I want to thank Rutgers University for all their assistance with developing our sampling protocols and for all the analysis they performed. I want to thank the Nature Conservancy for helping to fund this project. And lastly, my director for providing funding for this, but also giving me the flexibility to take on this additional work. With that, I can attempt to answer any questions you have.
Okay, would you like me to read you questions from the chat? Or you want to, is it easier for you to just try to do that yourself?
It's up to you.
Okay, I will ask you a
Okay.
pretty straightforward one from the chat, which is the most recent one, which is, what is the distance between station 701 and 704? And part 2, any thoughts about how much, if any, influence upstream sites are having on the downstream eDNA concentration?
Yeah, so the 701 to 704, basically you have 10 miles of river from the confluence to Paulina Dam. So 701 to 704 is about 9 miles. Yes, definitely there could potentially be influence of DNA from upstream locations traveling down, depending on the flow rates. I would think that would be more of an influence earlier on in the spring when flows are higher. As we get into summer and flows are a little lower, I'm not exactly sure how much of an influence those upstream sites are having on the DNA concentrations downstream, but it potentially could be.
Okay, Dylan, I see that you have your hand up. You want to go ahead and unmute yourself and ask your question?
Sure. Yeah. Hi. Amazing study. Really cool work. Thought it was especially neat how you were able to compare what's known about American shad spawning ecology with eDNA concentration. That was really cool.
Oh, thank you.
Yeah. So I had a metabarcoding question for you. Mostly, do you have your results yet? But also, if so, how did the sensitivity differ between qPCR and metabarcoding? Like, did you get full detections at all the sites you detected with qPCR? And then also just flipping through the Whittier paper, it looks like they just used total species diversity and not richness. And I was curious if you think that that'll end up being right for your IBI out there as well. And sorry, maybe this is too many questions all at once.
So I guess your last question about diversity and richness. So our main goal when we filed Whittier's paper, Bob Hughes and Whittier's paper, was to follow the methods they used, not necessarily all the metrics. We did test for species richness and species diversity as a metric, but it just didn't work for us. But we more or less followed the methods and types of analysis that they used. As far as the metabarcoding, I actually just received the results of the metabarcoding last week from Anthony Vestrano from Rutgers University. I barely had time to even look at it. I wanted to be able to put together a slide comparing our IBI results to the metabarcoding. Just wasn't going to happen in time. There was something interesting. So July 11th, our sampling event. So this eDNA 703 is the same station as our IBI monitoring PB55. So on July 11th, we noticed this pretty big peak in DNA. And then by July 18th, there's a huge decline. In between these two dates, July 11th and July 18th, on July 14th, we conducted our IBI monitoring. So kind of right in the middle in between. We did not collect any American shad young of the year through electrofishing, but through our metabarcoding, which again is not designed to detect fish which are rare or lower in abundance. We did detect American shad on 2 of the 3 filters we collected. So we have, 2 different methods. We have qPCR and we do have metabarcoding that are both detecting American shad. So I thought that was kind of interesting.
Can I ask one follow-up question?
Sure.
Yeah. So I'm curious if you once, I guess I'm just thinking or asking what you expect once you dig into the data, but do you expect that there are certain groups of fishes or aquatic organisms that will be better represented in the metabarcoding data than in the electrofishing, or vice versa?
Yeah, I'm not really sure. I guess I haven't got into it enough. Just looking back at the metabarcoding and just from what I know about the sites, it seems like the number of hits we have are very similar, it seems, to the most abundant species. Tessellated darter, black-nosed dace, they're the most abundant fish that we collected our IBI monitoring locations, and they typically showed up the most in our metabarcoding. As far as which fish potentially give off more DNA, yeah, that part I'm not 100% sure. That might be a question for Rutgers. They've been doing this work a lot longer. They've been doing a lot of monitoring for river herring. They may have a better answer than I do.
Okay. Thank you. Thanks, Dylan. There's another question in the chat that says, has water temperatures risen to and above 65°F earlier in the year in recent years? And if so, does this affect the phrenology of shad spawning?
I'm sure it has. Unfortunately, prior to this, I didn't really go back and look at, water temperatures in the Paulins Kill. Typically I'm more focused on the Delaware River prior to doing some of this eDNA work. I'm sure it does. The timing of it this year, I wish I had an answer for you. I mean, we have that information. I'm not sure. I think it probably did hit 65 degrees a little bit earlier this year because we had such low water. And I know my crew did observe some shad schools, I think maybe a week earlier than we did last year. So once I go back and look at the data, it's quite possible that spawning may have occurred a little earlier this year than last year.
Okay, question from Kevin, is eDNA less expensive than normal IBI sampling methods?
Well, I'm not sure how much IBI sampling actually costs. I mean, when you break down the salaries of all of our field crews, I imagine it's pretty expensive. So depending on what type of analysis you're performing, I think you're somewhere around $100 and $150 a sample for qPCR or metabarcoding. So that's one of the reasons that we're trying to collect metabarcoding DNA alongside our IBI results, is that we're hoping we can incorporate some eDNA into our monitoring so that it will not be as labor intensive. We won't hopefully need as large crews as we do. And, you know, maybe we can actually sample more sites than we currently do.
Okay, another one on here. Is there a website where this data is maintained? And I'm sure they mean available as well. That's Peter.
So our BFBM freshwater and biological website under fish monitoring is all of our IBI data is there. I do apologize. I don't think we've updated it since 2021. We're working on a new website. I'm not a web person, and I'm waiting for someone else to get the website up and running to get our newer data. We plan to add an environmental DNA section to that, but currently we do not have any of our eDNA data available. We barely have it into our database, and we don't really have a way of incorporating it into our national database store at WQX. So currently it is not available.
Okay, Alex is asking, I don't remember if you mentioned, was stream flow accounted for between eDNA samplings?
It was not. It was so we every day when we got to our first station, we did use the USGS gauge at just above Footbridge Park to record what the flow was. So we do have that, but we did not actually do any flow discharge measurements. It was something that, you know, I thought about early on when we were starting this work, but just for us to go out and collect just these 4 stations is pretty much a full day event, and adding a flow measurement would, you know, add considerable amount. So we do have what the overall flow was for each sampling day just from the gauge.
Okay, it looks like Peter's asking if you have that website available, can you put it into the chat?
Sure.
Looking for raised hands. If anybody wants to come off mute and ask their question, I think I covered the questions in chat, but if you put a question in there and I did not ask it, can you please unmute yourself and go ahead and ask? Okay, I have a question then, John.
Sure.
Well, I have 2 questions. One, early in your presentation, you showed a chart, sort of a table with ecological categories, responses for fish. And I'm wondering, you know, for macroinvertebrates, do you ever like look at the 2 side by side where you would be kind of figuring out, you know, like use them kind of to complement one another, or are they different enough that you're not doing that?
So we put together every couple of years, we put together an environmental trends report for the Division of Science and Research. And so that does have sort of a breakdown. The thing is, with macroinvertebrates and fish, you're kind of looking at 2 different biomonitors. One's looking at short-term stressors and one's looking at long-term, fish being the long-term. And they also monitor, they're very good at monitoring different types of stressors. Things like nutrients, macroinvertebrates tend to be a little bit better at detecting nutrient enrichment, whereas fish may be, you know, something else with water chemistry, dissolved oxygen, temperature, habitat impairments. So they sort of complement one another. There are situations where you will get a similar overall score, but there are also times when the score is different because one's kind of monitoring a acute change and one's monitoring a chronic.
Okay, and then I have the classic question for you, which is what was your biggest surprise? You know, either eDNA results versus IBI sampling, or just, you know, stuff that you didn't think you were gonna come across or be able to discover with the eDNA?
I was a little disappointed that we did not collect any young of the year. I think, you know, trying to figure out the timing, you know, because we collect these filters, they sit in our freezer for several weeks or months. They go to Rutgers, we get the results back, and I think I got them back in December for the qPCR. So, you know, we're kind of blind out there. We don't know for sure whether the fish are in there, and we're trying to time all of our monitoring around when we think the fish would be migrating. So I was a little disappointed we did not collect any young of the year. One thing I was kind of surprised by is, so we assisted the Pennsylvania Fish and Boat Commission, the co-op, with their young of the year seining. And we also collected DNA just prior to the seining, try to relate the DNA results to actual numbers of fish. And what I was surprised by was, on several dates, we actually had higher DNA levels in the Paulins Kill than we did on the Delaware River. Now, that could be a combination of effects that we're just not sampling deep enough in the Delaware because we're only going out so far, wading out so far, but I just thought that was kind of interesting.
Yeah, I mean, it seems unlikely that they'd be in the Paulins Kill but not spawning, right? I mean, it's just a matter of finding the proof or demonstrating the.
I'm pretty positive, just based on the video, you know, by the time you start seeing American shad adults in the water column, they're either spawning or they're post-spawn. Typically prior to that, you will not see them. They're in the deepest part of whatever river they're in, tributary, following the thalweg, and you just do not see them. And it's when they're actually up in the water column actively spawning or they're post-spawn that you see them. So I'm pretty sure that through our video that, you know, and the amount of DNA that we definitely have captured spawning, but until we probably physically collect either a larval or young of the year American shad, you know, we can't say definitively that they're there, but.
Okay, I see one more question which I can answer. What is the proposed date for the removal of the Paulina Dam? We propose to notch it this September. That notch would be really to begin to dewater the impoundment and to make sure that we can consolidate some of the sediment and we can lessen the impact of sediment moving downstream. So, you know, look for that notching, hopefully September, and then next year, sometime between July and September, we would be removing the entire structure. John, this is during the eDNA sampling, did you target specific subhabitats in the stream to filter water from, for example, pools or riffles?
For our IBI sampling, we did do that. So we tried to collect one of the filters at just riffles, one at just the pool, and one just at a run, if they were available in that reach. For our shad sampling, we pretty much targeted for about the time period we knew adults were in the river. We just targeted the main channel. We just collected all the water samples from the main channel as the year went on, and we're pretty confident that they're no longer adults and we're looking more for young of the year. We tried to target those side channels where they're more likely to find those larval fish and juvenile fish.
Oops, looks like we have time for one more question if there's one more out there. I can ask, if you have a question, you can just quickly unmute yourself. That's okay. Oh. Okay, hearing none, I'm going to remind folks that this will be recorded and you will find it probably in the next 3 or 4 days or so on njdams.org. Maybe Michelle, if you can throw that into the chat, you'll find all of the previous, this will be the 14th information session. You'll find all previous recordings there. I want to thank John for explaining this. This is some new ideas for everyone. I think we're all excited at your success and the things that you're able to tell so far. And I know for me, I am very encouraged and optimistic that this will really help us to tell the story of river restorations, not only dam removals, but all types of restoration. Michelle?
Just really quick, I don't know if it ended up in the chat, but where can people find any data that the DEP collects? So IBI or any of that stuff, even if it's not updated. Maybe you wanna shout
So
it out.
I did post our website in the chat.
Oh, you did? Okay, then I
So-
missed it.
That'll have our.
Don't mind me then.
Southern.
Yeah, it's up a little bit higher. Let's see. Yep, 150.
Okay.
Scroll back up in the chat.
Cool. Just want to make sure we had that. Thanks,
Okay,
guys.
thank you. Thanks. Okay. All right. Thanks again, John. And thank you everyone for joining
Thank you.
us. And keep your eyes open. We have another 4 or 5 of these planned before the end of the year. So,
Thank you.
okay. Thanks everyone. Thank you, John.
See ya.
Thank you.